All right. Wonderful. Well, welcome everyone. Thank you for joining us today. I have the pleasure of introducing Dr. Laura Zahodne, who is an assistant professor in the Department of Psychology at the University of Michigan. She trained in clinical neuropsychology at the University of Florida, the Alpert Medical School of Brown University, and the Columbia University Medical Center. Her research aims to understand how life course psychosocial experiences influence late life cognitive trajectories, the expression of neurodegenerative disease, and racial ethnic inequalities in Alzheimer's disease and related dementias. Her research group collects longitudinal and biopsychosocial data from large, representative, and diverse cohorts, including the newly established Michigan Cognitive Aging Project. She also co-directs the Michigan Center for Contextual Factors in Alzheimer's Disease, which is an NIA-funded research center for minority aging research that aims to grow the workforce and infrastructure for research on dementia inequalities. I'm very, very excited to hear your presentation. As a reminder, please add your questions and comments to the Q&A, and we will go over them at the end of the talk. So the floor is all yours.
Thank you so much for that introduction, and thank you for the opportunity to present today. I'm excited to share with you this aspect of my research program that addresses this really important public health and health justice concern of dementia inequality, largely from a biopsychosocial perspective. I'm going to start today by just giving some super brief background on racial inequalities in dementia. Then I'll share with you some of the studies that my lab has conducted to explore potential explanations for these inequalities that persist even after we control for some of the usual suspects in health disparities research. I also want to discuss a couple of challenges to this work. These challenges are not unique to research on dementia inequalities, but they seem to be particularly crucial to consider in this area. And then I'll end by talking about some of my ongoing data collection efforts in Michigan to minimize some of those challenges and to further this line of research. So I'm sure everybody here knows that the global population is aging rapidly. Which means that there are more and more people at risk of Alzheimer's disease. Right now, there are more than 6 million people in the US living with AD, and that number is projected to double over the next 20 years. But what pictures like these don't convey is who these people are. This disease, like many diseases, is not randomly distributed in the population. It differentially affects marginalized populations, including certain racial and ethnic minority groups. And just to demonstrate how disproportionate dementia rates are across racial and ethnic groups, I'm showing this study, which was led by Elizabeth Rose Mayeda, who used data from the Kaiser Permanente managed care healthcare system in Northern California to estimate differences in incident or new onset cases of dementia across these 6 racial and ethnic groups. And as you can see, African Americans who are shown in purple are more likely to develop dementia than other groups, such as non-Latino whites and Asian Americans. And while this particular data set was limited in its ability to identify the causes of these differences, a growing body of evidence for the social determinants of these group differences overwhelmingly shows that they represent a health inequality. So a health inequality is a particular type of health difference that is avoidable, unnecessary, and unjust. And importantly, these differences persist even after controlling for the usual suspects in health disparities research, namely socioeconomic variables like education and income, but also health variables, cardiometabolic diseases like diabetes, heart disease, diseases that have long been known to differentially affect marginalized populations. So what are some of the potential explanations for these differences that persist despite adjusting for those important known mediators? Well, first, we probably aren't routinely measuring all of the risk factors for dementia in our cohort studies. There may be risk factors that privileged groups simply aren't exposed to and therefore haven't been considered in research that uses largely white samples. Another possibility is that the dementia risk factors that we do know about might exhibit differential impact across race and ethnicity. That is, having a particular risk factor may be more or less risky for members of a particular group. So next, I'm going to show you some studies demonstrating both of these possibilities, starting with the first. So these next data come from the Health and Retirement Study, or the HRS. This is a large, nationally representative study of adults over age 50 in the US. That's run out of the Institute for Social Research at the University of Michigan. And for this study, we focused just on those HRS participants who were 65 and older at the time a comprehensive psychosocial module was added to the study in 2006. And in the study, we were interested in testing this specific theoretical model of psychosocial pathways involving reported experiences of discrimination. And the psychosocial variables we included were ones that previous research had hinted were important for minority cognitive aging, but had never been put together into a comprehensive model and examined as joint predictors of longitudinal cognitive change or systematically examined across racial groups. So here, discrimination captures the frequency of everyday experiences of interpersonal discrimination, which we conceptualize as a potent form of social stress. And qualitative studies have shown that these social experiences can result in changes in mood, as well as an increased perception that one is not really in control of his or her life outcomes. And we confirmed that this model fit the data better than alternatives, and we estimated all possible associations. But for simplicity here, I'm just showing those paths that were significant in the final model, and I'm not showing the demographic, socioeconomic, and health covariates. So compared to whites in the HRS, non-Hispanic Blacks reported more discrimination, which in turn predicted both more depressive symptoms and more external locus of control. And both of those psychological consequences of that discrimination were in turn associated with lower initial memory performance. But in addition to those pathways to initial memory level, discrimination in the study also directly predicted steeper rates of subsequent memory decline. So this study suggested that discrimination may be a central node within psychosocial pathways leading to worse memory for older Blacks. So that's an example of a psychosocial model of racial inequalities. In my lab, we've been trying to extend this work into a biopsychosocial model by investigating some of the physiological mechanisms by which psychosocial factors might get under the skin to affect health. So here, in this next study, we incorporated longitudinal biomarker data available in the HRS, specifically levels of C-reactive protein in capillary blood. So C-reactive protein, or CRP, provides a measure of systemic inflammation. And we know from prior work in both animals and in humans that inflammatory markers, including CRP, are elevated in the context of chronic stress. So this study used all of the available HRS data to test whether elevated CRP mediates longitudinal associations between everyday discrimination and episodic memory. Now, in this study, we didn't see a whole lot of intra-individual change in this 4 to 6 year period, so there was no longitudinal mediation. But we did see evidence for mediation within the baseline data, where we had a lot of inter-individual variability. So specifically, those participants who reported more frequent experiences of everyday discrimination had higher CRP, which was in turn associated with lower episodic memory performance. So some preliminary evidence that inflammation may represent one pathway by which racially patterned social stress could influence memory performance and ultimately dementia risk. Now, in addition to studying how the experience of discrimination gets under the skin, we're also interested in how it might get into the skull. So these next data come from the Washington Heights Inwood Columbia Aging Project, or YCAP. And this is a longitudinal epidemiological study that follows a regionally representative sample of older adults living in northern Manhattan. And about 1/3 of the YCAP sample identifies as non-Hispanic, Black, or African American. And this is the group I focused on for this study of discrimination and brain aging. So indeed, a subsample of non-demented participants in YCAP have undergone 3T MRI. And this study used available data from 2 MRI time points separated by about 3.5 years. So recently we added a psychosocial module to the YCAP to better understand racial ethnic disparities. And that module includes multiple measures of discrimination. And the inclusion criteria for this study included self-reported race ethnicity of non-Hispanic Black, the availability of at least one time point of usable neuroimaging and psychosocial data, as well as the absence of dementia at the time that the self-reported psychosocial measures were completed. So here's an image showing that Black participants who experienced more racial discrimination showed lower hippocampal volume at baseline. So this discrimination measure along the x-axis here queried specific experiences of racial discrimination in major life events at any time over the life course. It included questions querying discrimination in education, in the labor market, in housing, etc. So some example items were, have you ever been unfairly prevented from moving into a neighborhood because the realtor refused to sell or rent you the house or apartment due to your race? Or have you ever been unfairly denied a bank loan due to your race? We also found a longitudinal association between racial discrimination and white matter hyperintensities, which are an indicator of subclinical cerebrovascular disease. So specifically here, Black participants who reported more everyday discrimination showed faster accumulation of this pathology over time. So here, time is on the x-axis, and the 3 different lines correspond to model estimated trajectories for individuals reporting high, average, or no everyday racial discrimination. And as you can see, white matter hyperintensities increased over time among those individuals who reported high levels of everyday racial discrimination. Now, this is a different discrimination measure than the one that I showed on the last slide. This measure queried current day-to-day experiences of interpersonal discrimination. So example items there were, how often have you been treated with less respect than other people due to your race? How often have you been called names or insulted because of your race? And an interesting potential take-home message from this study relates to the fact that these 2 different measures of discrimination were associated with different brain outcomes, which might point to different mechanisms. So specifically here, the day-to-day experiences of interpersonal racism appeared to be most relevant to the ongoing accumulation of white matter hyperintensities, which might point to cardiovascular and inflammatory pathways implicated in chronic stress. In contrast, the accumulation of major experiences of more systemic racism across the life course appeared to be most relevant to baseline brain integrity, baseline hippocampal volume, which might suggest that the experience of racial discrimination in major life events has a stronger influence on brain and hippocampal development and maintenance leading up to late life than on atrophy during late life. And that baseline association could reflect known neurotoxic effects of stress on the hippocampus, but it could also reflect the long-term impact of systemic racism on life course opportunities and environments, which could affect brain health independent of stress. So just as one example, racial discrimination in housing could result in greater lifetime exposure to air pollution or other environmental toxicants that are known to be particularly deleterious to the hippocampus. So I think including multiple measures of discrimination can paint a broader picture about how social experiences might affect dementia risk. But recall that another potential explanation for these differences that persist, even after we control for the usual suspects, is that risk factors for dementia that we know about might disproportionately affect outcomes among certain social groups. And I'll talk about differential impact of two risk factors in particular, depressive symptoms and white matter hyperintensities. So for this next study, we used data from the norming study for the NIH Toolbox. This is a standardized set of computerized neurobehavioral measures that were developed through the NIH Blueprint for Neuroscience Research. And that norming study included people aged 3 to 85 living throughout the US. But for this study, we focused just on a relatively small subsample of participants who were aged 55 and older. And we found in the Toolbox norming study that even though Black older adults reported fewer depressive symptoms than whites on average, when depressive symptoms were present, they were more strongly related to lower cognitive performance among Blacks. And here's a picture demonstrating these different associations. And these results are consistent with literature showing that depression, which is often found to be less prevalent among Black adults, has also been found to be more chronic and to have a greater functional impact among Black adults. And this may be because non-Hispanic whites are more likely to have access to situational and environmental advantages and resources that buffer against the negative health impacts of depression. And conversely, membership in a minoritized group is associated with situational and environmental disadvantages that could compound risk. And we've since replicated this result in a couple different data sets. But this differential impact of risk factors appears to exist not just for psychological risk factors like depressive symptoms, but also biological risk factors. So here I'm focused on white matter hyperintensities, and I'm using data from YCAP. And we see that white matter hyperintensities are more strongly associated with worse cognition in multiple domains. So here I'm just showing a comprehensive language factor among Blacks than among whites in YCAP. And here again, we think that these group differences in the strength of association between the risk factor and the outcome could reflect an interaction between the pathology, white matter hyperintensities, and environmental variables important for cognitive resilience that are racially patterned. So in other words, advantages afforded to whites might not only protect against the accumulation of white matter hyperintensities, but might also buffer against their negative cognitive impact. So as you've seen up to this point, much of my research has focused on modifiable risk factors that help to explain racial inequalities in brain and cognitive aging. But the association between race, which is a complex sociocultural construct, and cognitive aging really reflects the sum of both risk and resilience pathways. And one implication of this is that the racial differences we're measuring in our cohort studies may actually be underestimating the health risks of social inequity, because different social groups also cultivate norms and behaviors that can be health promoting. So focusing only on risk can't tell the whole story. So I want to show you some examples of the benefits of looking not just at risk, but also resilience. So returning to the health and retirement study now, these are data to suggest that religious involvement may represent one naturally occurring source of cognitive resilience for many Black and Hispanic older adults. So this work was led by a recent graduate from our lab, Zarina Krall, who was interested in parsing this multidimensional construct of religious involvement into different components: strength of religious belief, frequency of attending formal religious services outside the home, and frequency of praying privately. And she examined how these three aspects of religious involvement differed across race, ethnicity, and related to memory. So here I'm showing just those paths in her statistical model that were positive and significant, and I'm not showing her comprehensive set of covariates. And as you can see, both non-Hispanic Black and Hispanic participants in the HRS report higher levels of all three components of religious involvement, consistent with previous research. And two of those components, private prayer and attendance, were in turn associated with better episodic memory. So even though these two racial and ethnic groups exhibited lower memory performance compared with whites overall, those disparities, shown in red, were offset by these independent resilience pathways shown in green. So I think this line of work really highlights the need to partner with communities to understand the cultural values, norms, and practices that are already in place, especially when we're thinking about intervention and prevention research. I'll show you another example now of the value of considering both risk and resilience in studies of racial disparities. This again comes from the Health and Retirement Study. This is a recent paper led by a graduate student in my lab, Emily Morris, in collaboration with a former postdoc at ISR here at Michigan, Dr. Lauren Brown. And they were interested in replicating and extending Dr. Brown's critical work on stress appraisal and mental health to cognitive health. And together they showed that Black older adults in the HRS reported being exposed to more chronic stressors than whites. And in turn, being exposed to more chronic stressors predicted worse cognitive health. So there was a negative indirect effect involving stress exposure that contributed to the overall total effect, showing a Black-white disparity in memory performance. But those Black older adults also appraised stressors as less upsetting than whites, showing evidence of adaptation that may reflect different historical and contemporary lived experiences and coping. And while appraising stressors as less upsetting didn't moderate the negative impact of stress exposure on memory, there was no interaction between these two stress variables, there was a main effect, such that appraising stressors as less upsetting independently predicted better memory. So similar to the set of results I showed you on the last slide, the Black advantage in stress appraisal offset the memory disparity, providing evidence for both risk and resilience among Black older adults. Now I want to spend some time talking about a couple of the major challenges to this work, which again are challenges to many types of research, but I want to hopefully show you that they may be particularly challenging to research on inequalities in aging outcomes. So the first challenge relates to selective survival. And this refers to the fact that socially disadvantaged groups, including African Americans, who are a focus of this particular paper, have higher mortality early in the life course. And what that means is that those who survive to late life and make it into our cohort studies exhibit greater physical and or mental resilience that enabled them to survive. So in other words, they're more heavily selected than non-Hispanic whites. And this leads to a narrowing of racial disparities at later ages, because you're comparing an increasingly selected African American group to a less selected white group. And we've shown this in both midlife and late life cohorts. The youngest members of your sample are likely to show the largest disparities, in part because there has been less time for this mortality selection to take place. But importantly, not only does selective survival affect the magnitude of group differences that we can observe in older samples, it also undermines causal inferences. And I personally love this empirical demonstration of survival bias that uses smoking. So this graph summarizes a lot of prospective epidemiological studies that have examined the association between smoking and incident Alzheimer's disease. So each circle in this graph corresponds to one study. And what you can see is that those studies that initially enrolled relatively younger individuals on the left-hand side of the graph, they showed that smoking predicted increased risk of Alzheimer's disease. But in contrast, those studies that enrolled relatively older individuals, the ones further on the right-hand side of the graph, they showed the opposite, that smoking predicted decreased risk of AD. Now, we don't think that once you hit age 75, you should take up smoking to ward off dementia. In other words, those negative associations aren't causal associations. Rather, if you survive to age 75 or 80 dementia-free, despite being a lifetime smoker, you probably have some sort of unmeasured, possibly genetic resilience. And the risk marker, smoking, becomes a proxy for that unmeasured resilience, and it can sort of masquerade as a protective factor. So selective survival is one reason why many studies, particularly those interested in dementia inequalities, are recruiting younger participants, in contrast to some more traditional dementia cohort studies where the mean age at study entry is around age 75. So selective survival is one challenge to disparities research and aging. Another challenge relates to recruitment. So a recent demonstration of the implications of recruitment for dementia disparities research can be found in this paper by Carrie Gleason and colleagues, who looked at Black-white differences in dementia conversion using data from NAC, the National Alzheimer's Disease Coordinating Center, which included over 30,000 participants from 30-some ADRCs throughout the US. And as you can see from this graph, when examining individuals who were initially cognitively healthy, there were, surprisingly, no racial differences in conversion to dementia. And similarly, when examining individuals who met NAC criteria for mild cognitive impairment, or MCI, at enrollment, Black participants were less likely to convert to dementia than white participants. So in both of these slides, the findings are in contrast to Dr. Maeda's study that I showed at the very beginning, as well as currently available meta-analyses of mostly population-based studies that estimate that dementia conversion is at least 60% higher in Blacks than whites in the United States. So if we take these NAC data at face value, we might conclude that there are no racial inequalities in dementia. But the authors of this paper note some important differences between whites and Blacks in the NAC dataset that suggest that these results might be an artifact of selection bias. So, for example, they note that increased efforts to diversify ADRC cohorts have led to divergent recruitment strategies targeting different racial groups. So while most of the white participants in NAC are presenting to these tertiary care clinics with memory complaints, most of the Black participants in NAC are recruited directly from the community. So just as one example, more white participants in NAC were referred by a health professional. And being referred by a health professional was in and of itself a unique predictor of dementia conversion. So the basic problem here is that divergent recruitment strategies can introduce selection bias. When the recruitment strategy, here community-based versus clinic-based, is associated with both the grouping variable, in this case race, and the outcome, in this case cognition. So as you can see in this figure, this is because clinic-based samples, shown in green or gray, are enriched for individuals who are at increased risk for AD. Whereas community-based samples, shown in cream, have more average risk for AD. And if white participants are disproportionately recruited from the higher risk clinic population, and Black participants are disproportionately recruited from the average risk community population, then we aren't going to be able to obtain valid estimates of racial differences in dementia risk. Now, this bias is difficult to overcome statistically, which really highlights the need for new studies that were explicitly designed to examine health inequalities, as opposed to retrofitting existing studies of mostly white people to begin to address health disparities questions. And when I came to the University of Michigan, one of my goals was to launch such a study that was explicitly focused on health disparities. So in 2017, which feels like only yesterday, we started recruiting people into the Michigan Cognitive Aging Project, or MCAP, which now follows a regionally representative, racially balanced cohort of adults transitioning into late life. Participants are recruited from Washtenaw and Wayne counties here in Southeast Michigan. Here I'm showing you the location of the University of Michigan in Ann Arbor, as well as the city of Detroit, where many of our participants live and work. So at MCAP, we use a direct mail recruitment strategy that takes advantage of sociodemographic data from the US Census and mailing addresses from the 2016 voter registration list. So our initial address-based sample included over 5,000 adults aged 60 to 65. We were really excited to see a response rate of more than 10%, which is really twice that of the response rate that's typically reported in the literature for this type of direct mail recruitment strategy. And in the background of this slide, you can see the racial distribution of residents in the Ann Arbor, Ypsilanti area. So in this image, whites are shown with blue dots and blacks are shown with green dots. And these are the kind of data that we use to guide our sampling strategy, which led to us only targeting census tracts in the city of Ypsilanti on the right-hand side of the screen for our Washtenaw County recruitment and the city of Detroit for our Wayne County recruitment. So we wanted to maximize the representativeness of our sample, so we implemented minimal inclusion and exclusion criteria. Now, we initially planned on limiting enrollment just to individuals in the address-based sample. But along the way, we chose to respond to our enrolled participants' overwhelming requests to refer people that they know. Not only did we want to be responsive to participant preferences and to foster mutual collaboration with the community, we also saw this as a way to systematically study recruitment and enrollment factors. So individuals who ended up being recruited via this chain referral method are clearly identified in the data set. And interestingly, our preliminary analyses show that those referred participants have lower socioeconomic status and poorer health than the original address-based sample. And that finding is consistent with the original conceptualization of chain referral recruitment methods as a way of recruiting participants from populations that have been historically underrepresented in research. So we also wanted to eliminate one of the major barriers to research participants. So we allowed participants the option of being seen in their homes. So in our study, 1 in 5 participants opts for a home visit. And we think that option has helped us to achieve a more representative sample. Related to that is that our biomarker sub-studies are optional. So far in MCAP, about 83% of participants have agreed to provide blood samples when asked, and about 67% of participants have been both willing and eligible for neuroimaging. And we envisioned MCAP as a longitudinal study from the beginning, so we're particularly invested in retention. So for every participant, we collect contact information for at least one individual who would be expected to always know their whereabouts and who doesn't live in their household. We also create and send annual newsletters to maintain contact with participants. It also encourages updating of contact information through the US Postal Service. We've just finished up the first follow-up visits with participants, which take place 2 years after enrollment. And so far, our retention of survivors has been close to 85%, which we think is pretty good for this population. So this is a really busy slide with basic information about the first 500 participants enrolled in MCAP. The pie chart on the upper right shows the racial ethnic breakdown. And as you can see, MCAP is largely a racially balanced sample of non-Latino Blacks, shown in orange, and non-Latino whites, shown in blue. We are aiming for a racially balanced sample rather than mirroring population demographics. And this is because we want to maximize power for our between group analyses and also for our within group analyses. So far we don't see racial differences in basic demographics, but we do see moderate to large socioeconomic disparities across all metrics. And we also see moderate to large physical and mental health disparities. But really the goal of MCAP is to look at cognitive disparities. So here you can see the Neuropsych Battery. It can be summarized adequately by these 5 latent factors. A lot of these tests were chosen to harmonize with other studies, including NAC, with some changes like using color trails instead of the traditional trail making test to accommodate lower literacy levels. And in addition to these paper and pencil tests, we also give most of the cognitive measures from the NIH Toolbox, including their reading test. So one of the primary goals of MCAP is to advance understanding of cognitive disparities. Here you can see those disparities prominently in the unadjusted data. So on average, Black participants score more than 1 standard deviation lower across our battery compared with white participants. And those disparities are somewhat attenuated in the demographically adjusted data. So this graph controls for age, sex, and most impactful educational attainment. And through our ongoing projects, our lab is dedicated to more fully characterizing the pathways that create and sustain these disparities, with a particular focus on psychosocial factors and their biological mechanisms. So that's a little bit about MCAP, which is one new cohort that I'm excited about. Another new cohort study is the Detroit Aging and Memory Project. Through this project, we were recently funded to obtain the very first dementia prevalence estimates among older Arab Americans, which is a group that has been virtually absent from research on dementia and many other health outcomes, due largely to the fact that the US government legally classifies Arab Americans as non-Hispanic white. Though there has been evidence that Arab Americans have been increasingly racialized and subjected to various and unique forms of interpersonal and institutional discrimination since 9/11 and the initiation of the War on Terror. So this region that's just west of Detroit has long been home to the most concentrated and visible population of Arabs outside of the Middle East. So here I'm representing the city of Dearborn, Michigan, with the logo for the Arab American National Museum, which is located there. So DAMP has undergone a painful COVID delay and is scheduled to go into the field this year using state-of-the-art sampling and recruitment strategies that were designed with the Survey Research Operations Group at the Institute for Social Research at the University of Michigan. And our goal is to recruit a population representative group of Arab Americans aged 65 and older to ascertain dementia prevalence using a battery of cognitive and functional measures that we translated into Arabic with the help of many, many community partners. And based on evidence for the high prevalence of dementia risk factors like diabetes and depression among Arab Americans and the limited research that's been done, we think that there may be disproportionate rates of dementia in this group as well. And making those disparities visible would highlight the need to disaggregate this currently invisible group from non-Hispanic whites, which very nearly happened with the 2020 census when recommendations to add a Middle Eastern, North African, or MENA demographic category were unfortunately not ultimately enacted. That change could have really facilitated high quality research on health inequality and other types of inequalities, and hopefully will happen with the next census. But in the meantime, we're invested in this primary data collection in order to disaggregate this community. So talking about facilitating research on health disparities. I'm also super excited about our relatively new NIA-funded Resource Center for Minority Aging Research in Michigan. This is the Michigan Center for Contextual Factors in Alzheimer's Disease. And our center focuses on context. Some of our center activities that you might be interested in include hosting a summer data immersion program where scholars from across the US can come to Ann Arbor to learn about how to leverage public data resources for research on ADRD and minority cognitive aging. We also have a competitive enrichment program that provides mentoring and pilot funding to junior faculty or mid-career faculty who wish to transition into research on dementia inequalities. So keep an eye out for our annual requests for applications for these programs. Some of our other center activities include developing participant registries to facilitate AD research, both with Arab Americans in Southeast Michigan, as well as Latinos in West Michigan. And through those efforts, we hope to develop the research workforce and the infrastructure for research on dementia inequalities in Michigan and beyond. So I want to end now just by acknowledging all of the participants in the studies that I work with, not all of which I was able to present on today. And I also want to thank my incredible collaborators at the University of Michigan and at Columbia University, and generous funding from the NIH and the University of Michigan. And I want to end with this call to action and an image of our virtual HERE lab group that's based in Ann Arbor. And thank you for this opportunity and for your attention. I'm looking forward to your questions. Thank you.
Thank you so much for sharing your work with us today. So if anyone has any questions, please feel free to add them to the Q&A. And while people write their questions, I actually have a question. So I think the data that you showed related to stress and cognition is so interesting. And I was wondering if you have explored, you mentioned religion and some protective factors, and I was wondering if you've explored any lifestyle factors such as social engagement or physical activity work that could help dampen the negative effects of stress on cognition.
Yeah, that's a great question. Yeah, so I think another major aspect of my research program is about social relations. So I'm very interested in kind of disentangling this kind of general effect that social relations seem to be good for cognitive aging. We saw that, in the Livingston article that summarized what are the major modifiable protective factors for dementia. Social isolation was one of them. But social relations is such a complex, multidimensional construct. What is it about social relations that it seems to be most impactful for cognitive aging? So we do a lot of work to try to disaggregate that construct. And so what we have found is that it's not really the number of individuals in your network that seems to be important for cognitive aging. It's more the frequency of contact with those members. So the more frequently you have contact with your network members, regardless of how many are there, that seems to be good for cognitive aging. We're also interested in kind of who is it in your network that seems to be most beneficial. So we've looked at the composition of social networks in terms of kin and non-kin. And we have some converging evidence from a lot of different data sources that friends in particular, or non-kin, seem to be most strongly related to cognitive outcomes. We just had a recent publication in the Alzheimer's and Dementia showing that having more friends in your network buffered the negative impact of brain pathology on cognition. So this was a paper that sort of replicated and extended an early paper from the Rush group showing that larger social networks buffered against neuropathology measured at autopsy and cognitive performance later in life. So we showed using in vivo neuroimaging a similar finding that bigger networks were important, but we showed that it wasn't just a bigger network, it was who was in your network, and it was strongest for number of friends. So having more friends in your network seemed to be most beneficial. And we've been thinking a lot about the mechanisms Yeah, the mechanisms underlying that, why is it that friends would be most beneficial? So one idea is that our relationships with friends, particularly later in life, if you think about socio-emotional selectivity theory, we tend to prune our social networks as we get older, such that the friends who remain in our social networks are really the ones that we have the most high quality relationships with. So compared to family, where sometimes we have ambivalent relationships because these are kind of required relationships, friends seem to be more unidimensionally positive. So it could have to do with affect and well-being, but it also could have to do with what behaviors you're doing with your friends. So, you mentioned physical activity is another thing. So there's daily diary studies showing that when people are around family members, they're more likely to be doing passive activities or household activities. When they're around friends, they're more likely to be doing challenging, cognitively stimulating activities. So we have a paper looking at leisure activities, physical and cognitive leisure activities as potential mediators of having more friends in your network. So we think that we're trying to break down this finding that social networks are good into more actionable targets for interventions.
That is so interesting. And do you hypothesize that this will be different depending on cultural background, race and ethnicity?
Yeah, that's a great question. Yeah, we do have one paper in YCAP where we looked at the composition of social networks and the number of individuals in a social network in whites and blacks, and showed that these effects were stronger for blacks in YCAP than whites. So all the more reason why we think this is an important avenue to go down, not just for healthy aging in general, but for health disparities in particular.
Thank you. And Paula, it seems like you have a question.
Yes, I do. Thank you so much, Dr. Zahodne, for this fascinating presentation. It was incredible. I was curious, being that you have access to such culturally different groups in your different studies, have you looked at the impact of family as sort of a specific social support system, and how maybe family factors, including family structure and different aspects like that, might vary by culture, and therefore sort of differentially be a protective factor among these groups, for dementia risk?
Yeah, that's a great question too. So, we do see differences in social network structure across groups, as well as differences in the strength of associations between certain social relations variables and outcomes, which I think is really important to take into account when we have these really diverse data sets and when we're interested in health disparities. And it may be that different aspects of social networks may be differently protective for different groups. It may also be that different aspects are differently protective at different stages. So most of the research I'm talking about is in normal cognitive aging. So before people are diagnosed with dementia, we've thought a lot about how the fact that once people start to experience chronic illness, disability, dementia, their social networks change and also their needs might change as well. So that might be where having stronger family relationships come into play, because we know from caregiving literature that caregivers are usually coming from the family network and not from the friend network. So I think that's another place where we really need to look at cultural differences in order to understand what the care network availability is and how social networks can absorb the responsibilities that come with dementia caregiving too. We're kind of looking at the later ends of the spectrum.
Thank you.
Any other questions?
I have another question. So related to your newer study looking at it's primarily Middle Eastern or Arab immigrants to the United States. Will you be taking into consideration sort of timing of the stressful events? So many of the groups that you're covering already, that it talks. There's so many systemic and ongoing stressors and inequalities that have been going on for centuries. And then, but I also think about, this particular group, and like you said, the impact of 9/11 and sort of everything from there on. So how are you considering that in studying this group?
Yeah. So I think it's important to collect as much data as we can to capture those experiences. So we do take detailed histories of their immigration history. So how old were they when they immigrated, what year they immigrated, so that we can match those timings onto not just events in the US, like the Muslim travel ban, but also events in their home country in terms of political turmoil or war, and also reasons for immigration. So, one kind of pattern of results that seems to be emerging for Arab Americans is that we don't see the healthy migrant effect as strongly as we have seen for historical research on Mexican Americans, for example. And, one reason for that may relate to reasons for immigration. Historically, Mexican Americans were migrating more for economic opportunity, so more pull factors, whereas Arab Americans more recently have been immigrating more for push factors, fleeing political instability in their home country, for example. And so in the pull factor scenario, there's more likely to be selection pressures, that the healthiest people are the ones who are migrating. But in the push factor scenario, we don't see that selection. So it's more representative people who are migrating, and they're also going through trauma, which we know has health impacts. So timing is critical. And I also think that a lot of the historical literature about migration and health, is limited in that it focuses mostly on Mexican Americans, but also that it focused on more historical samples that had more homogenous reasons for migrating. I think today, even the Latino immigrants we're seeing are immigrating for a lot of different reasons, including refugees. So I think sort of the knowledge base about immigrants and health, it really needs to expand to account for heterogeneity in who's migrating and why and from where. And that's going to give us much better understanding of how immigration experiences may be relevant for health and how we can target interventions toward those who might need them most.
Thank you.
Thank you. We have two questions in chat. The first one is, can you speak to neighborhood characteristics and what your work has shown, how, if this relates to experiences of discrimination?
Yeah, that's a great question. So we are very interested in neighborhood characteristics. So one thing I'm really excited about in MCAP is that we have now, through a K Award, a former postdoc in my lab, Kathleen Soul, who's now a research investigator at ISR, had a K Award that allowed her to geocode all of the current residential addresses for participants in MCAP. And she's been able to access some really rich data about neighborhood characteristics through NANDA. NANDA, N-A-N-D-A, is National Archive of Neighborhood Data. And it's a publicly available data resource through the Institute for Social Research at the University of Michigan that aggregates across multiple data sources to tell you things about the environment, like how many healthcare providers are in a neighborhood, what is the SES of the neighborhood, how many social services are in the neighborhood, what's the air quality in the neighborhood. So we have now connected with a lot of those variables and are looking at those variables that seem to be really important for residential segregation, that show, why certain neighborhoods may have influences on health above and beyond individual risk factors. One thing that has really emerged as an important variable that it wasn't necessarily what we thought would emerge is broadband internet access. That seems to be a really sensitive indicator for residential segregation that also seems to be super important for cognitive health, and is a pretty policy relevant variable that can be modified with policies and interventions. So your question, though, was about discrimination and neighborhood characteristics. And that kind of makes me think about the concept of ethnic enclaves and how results that we may find within samples recruited from an enclave may differ from samples who are recruited from other places. That's something that we think a lot about in our Arab American data collection, because we originally planned just to recruit within the ethnic enclave, and we have since kind of thought about expanding recruitment outside the enclave, because people may have different experiences within the enclave and may have access to different resources within enclaves versus out. I'll also call out research in African American aging that has shown that individuals living in higher SES context, Black individuals living in higher SES contexts, report more discrimination than individuals living in low SES context. So I completely agree that there are differences in experiences as a function of neighborhood, and there may be interactions between these variables we think are important, like discrimination, and the neighborhood or socioeconomic context in which they happen. The higher SES context may provide some resources, but it may have sort of the additional risk factors in terms of exposing individuals to more discrimination and more prejudice.
And we have a question from Rebecca Mariglio. Can you speak more to differences in studies that show steeper cognitive decline in African Americans versus those that show baseline differences but not differences in decline? What factors do you think could account for differences in these findings across studies?
Yeah, that's a great question. And I think I'm going to circle back to something I talk a lot about, which is recruitment. So in YCAP, for example, we do show that there are subtle racial differences in rates of cognitive decline, such that the African American participants are declining slightly faster than white participants. But there are also reports from the Rush studies where Black participants in the minority aging research study show less cognitive decline than whites in the Memory and Aging Project and the other Rush studies. And so I think one reason for that could be recruitment. So whether individuals are recruited using an address-based sample versus more convenience sampling. One way we've tried to quantify that before, to quantify the concept of representativeness, is by looking at the average education level of a sample and comparing it to census data, which tells us about the average education level for that population in that region. So, we found in YCAP that difference is really small, that the education level of Black participants within YCAP is very similar to the education level of Black participants living in Northern Manhattan, which provides, we think, some evidence that this is a representative sample. Other studies, the education level is much higher than that of the surrounding population, which might suggest there is some selection bias. So I think selection bias related to recruitment is one reason for that. Another reason is selective mortality. So if we start studying people later, we're studying the most resilient or the healthiest of the population that have, in many cases, exceeded their average life expectancy for their group. So we could be seeing this resilience that may be an artifact of selection. And there might also be selective attrition after the fact. So people who are most likely to decline or most likely to drop out of your study, you may not be able to detect decline as readily. And so all of these issues kind of come with various options for handling them statistically, whether it's through inverse probability weighting or joint modeling of cognitive trajectories with death, that can get us closer to a causal inference for what is the effect of these experiential factors on rates of cognitive decline.
And then we'll take one more question. Talia has a question.
Hi, sorry, I'll try to be quick. I don't know if I may have missed if you addressed this, but you talked about having the equal representation of Black and non-Hispanic white in this sample in Michigan, and I'm not sure what the demographics of your Black sample really allow for, but I was curious if you have any goals of investigating differences across culture within some of your Black samples. So looking at Black families or Black participants, either of kind of different nation of origin or different cultural background regarding being Caribbean descent or more recent immigration status from African continent, or if you have plans to look at any differences across those groups.
Yeah, that's a great question. So we do have information about where people were born. It's a very low proportion within our sample of Blacks who were born outside of the US. But there's quite a bit of variability in where within the US they were born. They were born in the South versus the Midwest. And so we do have some projects right now that are characterizing the childhood experiences of our Black participants. So we're looking not just at what region of the country they were born in, but what types of schools they went to. So we ask about the racial composition of their school, which because of residential segregation and how public schools are funded can be a proxy for school quality. We also ask about whether they went to a public or a private school. We have them rate the quality of their school. So we're seeing a lot of within group heterogeneity in these early life factors that are predicting cognitive performance much later. So I think this is an incredibly important point, both the point about looking at heterogeneity within the African American group and the point about considering life course experiences that may be relevant for cognitive aging. And then I also just want to say one more thing about the previous question in terms of level versus change, just to reiterate that the racial differences we see in initial cognitive level are much stronger than any racial differences that we see in subsequent rates of cognitive decline. You know, some studies show that they're there, some studies show that they aren't, but they're always very small. So when we talk about racial differences in dementia, the initial level of cognition is much more important than the rates of change. So these dementia inequalities are really reflecting life course opportunities and experiences much more than just zeroing in on late life. It's not that this is explained by greater late life pathology. This is much more strongly influenced by life course opportunities and sort of where people are when they enter late life, because meeting criteria for dementia just means you fall below a threshold, and you can get below that threshold by starting at a lower point or by declining faster. And so for dementia disparities, it's really the level difference that seems to be most important, which again highlights the importance of taking a life course approach and not just honing in on 65 plus.
Well, thank you so much for the wonderful presentation and for answering all of our questions. Yeah, well, I hope you all have a great weekend, and we hope to have you in person in the future.
Well, thank you so much for the opportunity and for your questions. Have a great weekend.
Thank you.
Bye,
Thank you so much. Bye.
Thank you.
Bye.